Hello everyone, I'm Zhi-Jie Cao from Gegao's lab at Peking University, China. Welcome to my poster presentation, where I introduce you to our recent work on querying heterogeneous single-cell transcriptomics datasets via adversarial learning. Single-cell RNA-seq technologies have been evolving rapidly in the last decade. As the technology matures and becomes increasingly accessible, more and more data are being produced. This slide shows a common data analysis pipeline for single-cell RNA-seq, which involves annotating the cells in terms of cell types or differentiation trajectories. Conventionally, the data annotation process involves multiple steps and various hyperparameters to tune, guided by prior biological knowledge, which undermines the comparability across studies in many cases. With a vast amount of annotated data already there, it is now possible to annotate newly produced data by querying the existing data as a reference. However, the biggest obstacle of cross-dataset single-cell RNA-seq querying is the so-called batch effect. Generally speaking, batch effect can be caused by various factors, like the difference in experimental procedures and sample conditions. As a result, when we analyze multiple datasets profiling the same sample type, the cells would form clusters not only based on biologically relevant cell types, but also by individual datasets, which would confound the estimation of cell-to-cell similarity when performing cell querying across datasets. Here we designed CellBlast, a single-cell data querying method to solve this problem. CellBlast is based on a deep learning model that learns low-dimensional cell embeddings from the gene expression data. The model is based on adversarial autoencoders, also called AAEs. Similar to the more commonly used VAEs, it projects high-dimensional gene expression data to low-dimensional latent space via an encoder neural network, from which the original gene expression data can be reconstructed. The AAE model is extended by batch adversarial alignment. Specifically, an additional batch discriminator network is introduced to predict the batch origin of individual cells based on cell embeddings, and the encoder network is trained in an adversarial manner to fool the batch discriminator. So when the model converges, cell embeddings of the same cell types from different batches would be aligned together. This is the same example we showed before. After applying the model, we see that the different datasets are aligned pretty well, while the different cell types are still clearly separated. In a quantitative benchmark, the CellBlast model also compared favorably with other batch effect correction methods, in that it achieves overall the best dataset alignment as measured by zero alignment score, while maintaining high cell type resolution as measured by cell type mean average precision. One advantage of using AAE over VAE is that the posterior distribution of cell embeddings of AAE is implicitly parameterized by the encoder neural network. This allows the posterior distribution to be super flexible. We found that due to the flexibility, the AAE posterior is able to capture the local manifold structure. For example, we see that the cells on the border of the two cell types actually have their posterior distributions extending in roughly the same direction as their respective cell clusters. Since each and every cell has a posterior distribution, we thought this can actually be utilized to improve the cell-to-cell similarity metric. Here's an illustration of how it would work. Well, we also have two cell clusters. If we measure the cell-to-cell similarity with Euclidean distance, some cells of the same cell type would receive a larger distance than cells of different types. Apparently, that doesn't make sense, but if we rescale the local space to make the posterior distributions isotropic, distances in the rescaled space make much more sense. We call the rescaled distance NPD, and we indeed observe that using NPD resulted in an improvement of cell type resolution in real data. We applied CellBlast to an example of rare cell type discovery. Ionocyte is an extremely rare respiratory epithelial cell type, which was discovered by two independent studies using 10x and InDrop, respectively. We artificially removed ionocytes from the 10x datasets and used it as a reference to annotate the InDrop datasets. We hoped that CellBlast can correctly annotate all the other cell types, while also identify the existence of this unknown cell type. Indeed, CellBlast accurately annotated the majority of the cells, and there was a small group of cells rejected because their NPD were not small enough to reach statistical significance as quantified by empirical p-values. Cluster analysis of the rejected cells revealed that cluster 3 has the largest p-values, meaning that they are the least similar to reference cells and very likely a new type. We went back and checked the original cell type annotations and found that these were indeed ionocytes. Along with Cell Blast, we also built a well-curated single-cell RNA-seq reference database called the Animal Cell Atlas. We annotated all cells with cell ontology, which is a structured, controlled cell type vocabulary, so that the annotations can also be comparable across datasets. We also provide an easy-to-use web-based cell querying portal based on Cell Blast and ACA, so check it out if you're interested. And thanks for your attention. If you have any questions, feel free to contact me either on the conference platform or via email or GitHub. Thank you.